I would like to introduce you to analyzing the animal samples which have been performed in our animal trials on a more in vitro and omics approach. Therefore, as you have seen before, to introduce you again to our aims, we would like to screen for unintended effect of the Bt-maize material by use of alternative techniques and approaches, and therefore we added some new and well-known in vitro assay systems. The task is to perform animal omics studies, and another task is to provide new data on possible in vitro cell culture systems. This is our strategy we followed, and we put on the left side the intestinal tissues for RNA extraction, liver for RNA and protein, and wet samples for metabolites and immunoglobulins. We use these technologies called NGS or RNAseg, next generation sequencing. It's a deep sequencing approach of the intestinal tissue we selected. And we furthermore, in parallel, use the targeted approach using an RT-PCR assay system, it's so-called array. For liver, we use microarray systems for detecting RNA as well. Furthermore, targeted analysis with our qPCR array system and a proteomics approach using 2D expression scanning for proteins. For the wet samples, like serum or urine, mass spectrometry and NMR techniques are used, and as well immunoassays. We would like to obtain a holistic and as well a single targeted approach for transcripts, proteins, metabolics, and a specific pathway reflecting potentially the normal or disturbed cellular cell functions. On the second approach, we used known commercial intestinal cell cultures from rats or a new established porcine cell culture, and looked for vitality of these cells using several different assays, a phenotypic characterization, and another online system which is called Intelligence, which is looking for single cell reaction online. And finally, we used proteomics approaches as well here to get out potentially with the effects of GM-maize on cellular processes measured in vitro systems. So I would like to highly condense now our results. We have generated within the last 2.5 years in our labs, and therefore I can only show overviews here, no single results in these 10 minutes here. To summarize our holistic approach, a strong work relation of circadian clock genes have been analyzed if we have looked to the intestinal liver tissues from our 90-day studies, A and B, and the 1-year study. This is a different approach. So we only see in this study design that we are directly depending on the time of sacrifice, that we see some regulated genes there by using the RNAseg and microarray data. When we use these targeted approaches using the PCR arrays, the cell signaling pathways we revealed were not depending to GM feed, but we inspected transcription genes of apoptosis, unfolded protein response, DNA damage, and repair. But we couldn't identify any of these more than 60 single genes which have been looked for expression changes. We could not see any effect directly shown for GM feeding. So if you look to the 1-year study in the last year, we could identify ileum tissue for RNAseg and qPCR data, and we could only find minor effects on the circadian rhythm because we were able to correct these study designs of this 1-year study. So the effects are not as high as in the 90-day feeding study. But until now, we couldn't identify significant GM effect concerning this study, and this is now comparable to the 90-day studies until now. You can see here a so-called volcano plot, and you can see only these effects which are here, which might be significant. If we look to the proteomics of these cells, we found that the intestinal slices, there was a high variation, that we could not find any line how to equilibrate this, probably due to the microbiota which are highly changing between the individuals. So the 2D method, this 2D electrophoresis system to extract these patterns indicated here as one example are not applicable on intestine slices. Anyway, in the rat livers, this problem didn't occur, but there were not found any GMO-related effects. But as seen previously for the expression analysis using the RNA, circadian clock genes were as well affected in the liver proteome of study A and B. As a control experiment, we can now indicate and look for the specific one-year study, because we know that we have some mycotoxins in this study contaminating under the threshold of our feed. We found in the 2D approach some spots which have been dysregulated due to feeding of the mycotoxin contaminated diet, but not related to the GM. Switching now to the metabolomics approach, no major metabolomic effects related to the MON810 cultivars can be detected in these approaches. Relevant effects of the different controlled factors are more or less based on the following order: gender first, then study A and B, and finally Bt-maize doses in this correlation. And the resultant metabolomic networks are in coherence with coordinatedly regulated nutritional physiology in our system. Finally, the allergenicity and digestibility test showed that there is no Qui-1ab specific antibody detected in any rat serum of our studies, but slightly differences have been observed in maize in between the isotypes, but no correlation with the genetic transformation, especially no Qui-1ab content. If we look to this Qui-1ab digestibility, as seen here, was one example. This was found to be relatively robust, and we could test these on whole floor, and it did not evidence significant effect of the genetic modification, especially on the intrinsic allergenicity of the maize, which have been used here in our studies. Finally, now the cell culture systems we have introduced, we could not find any significant GM-related effect in these cell cultures, especially in the rat intestinal cell system. The treatments of the animals could not be directly correlated, the in vitro with the in vivo system. So this is a major point of our approach. If we see any in vivo effects, we could probably better correlate this with in vivo. But in both systems, we could not find any significant effects. Potentials to establish this and other new cell culture systems are obvious. And the evaluation of the combinatory tests, which may be additional test systems for these approaches, may be worked out in the future. Exposure, and this is our main interesting result. Exposure of up to 50% of flora or maize material or plant material can be exposed without detrimental effects on these cell cultures. Let me now conclude and recommend these findings. In the omics studies, we could show that in case of targeted approaches, the integration of omics analysis might be a potential tool for identification new pathways, if there are any pathways underlaid by these approaches, and might therefore provide a mechanistic tool for decision making. The disturbance of pathways of toxicity in our system have not been detected in the 90-day and 1-year study until now in the 1-year study. This is going on. The longitudinal system, which have potentially the potential to detect progressive changes when compared to single endpoint measurements, and the GRACE data underline that the application of omics approaches without a targeted and testable hypothesis, as well adequate controls, neither facilitated the hypothesis generation nor did it reveal unintended effects related to the feeding of MON810. Our GRACE data indicate furthermore that omics analysis are able to detect experimental variations, e.g. the time of sampling, which made our expression now possible to directly correlate the time of sacrifice to expression changes. The experimental design should be optimized always for such approaches because they are very sensitive to detect here changes. The validation of novel in vitro assay systems, as introduced here in our GRACE program, showed that aqueous plant extract can be applied up to 50%, but this requires a comparison to in vivo data, and these are not available until now. So we don't see effects in vivo, so we will not find effects in vitro like in our approach. Targeted feeding trials with whole food and feed leading to toxicity are needed to prove the validity of the developed RAS systems here. Currently, there are no in vitro methods for complex endpoints like repeated dose systemic toxicity, and the development of such assays will require major scientific advances in the future. Therefore, in vitro assays for specific endpoints of toxicity can be developed. Corresponding positive controls need to be selected, and this shows up in our 1-year study, which are partly contaminated by mycotoxins. Therefore, we may measure some changes. Now, the final conclusion is that immunotoxicity testing can provide extra information, especially in the case of new proteins, which might be incorporated in the diets for human and animal nutrition. And safety concern basically on this initial in vitro and in silico assays can be analyzed. Currently validated standard operation procedures for a number of in vitro immune function assays are still not available, and they might be worked out in the future. At the moment, different in vivo test systems using animal models are available, but have not yet been validated for regulatory processes. At high inter-individual variability in animal immune responses was observed and might limit the applicability of the used assay systems. Finally, I would like to acknowledge our co-workers and thank you for your attention.